Today we are going to be talking about transcription in prokaryotes. To begin, we need to look at the promoter region of the gene. This region is upstream of transcription and is involved in the binding of RNA polymerase prior to transcription. Within the promoter region, we have two consensus sequences which RNA polymerase identifies to bind to the gene. These include the -35 region and the -10 box. However, RNA polymerase also needs the sigma factor to bind to it to create the hollow enzyme. Then RNA polymerase can bind to the promoter region of the gene. The +1 region of the gene signals the start of transcription, and this is the point where elongation of transcription begins. Also, the sigma factor needs to detach from RNA polymerase to allow it to move downstream of the gene. The function of RNA polymerase is to synthesize RNA as it moves downstream the gene being transcribed. It also unwinds the double-stranded DNA molecule and closes it back up as it moves along. If the -10 box is deleted from the gene, then RNA polymerase can no longer bind to the promoter region of the gene, and therefore transcription cannot occur, and so the enzyme would not be able to metabolize sugar because it could not produce the necessary proteins in order to do so. During elongation of transcription, RNA is being built using a DNA strand. The DNA strand that is used to create RNA is called a template strand. This strand must be in the 3' to 5' direction, because RNA polymerase can only add RNTPs to the 3' end of RNA. This means that RNA is in the 5' to 3' direction. The other DNA strand is the coding strand, and this strand is identical to the RNA strand being built, except there is a switch between thymine and uracil. This strand is antiparallel to the template strand and is in the 5' to 3' direction. RNA is built by adding to the 3' end. This is where the phosphodiester bonds are created between each nucleotide that are added by RNA polymerase. This continues until termination. It's like puzzle pieces. Termination includes ending the creation of RNA. One way for RNA elongation to terminate in prokaryotes is to form a hairpin loop, also called termination hairpin, which signals RNA polymerase to stop. This forms as a result of inverted repeats of RNTPs, as shown here, with the RNTPs back-to-back binding forming this hairpin loop. This is followed by a long repetition of adenine called poly-A tail on the template strand to create a long repetition of uracil on the RNA strand. The bond between adenine and uracil is relatively weak, and this allows for the RNA strand to break apart from the DNA strand. This type of termination is called row-independent termination. Finally, we have our RNA strand made from transcription. The whole point of creating this molecule was to create a copy of DNA to use to later code for proteins. This RNA molecule is then sent to a ribosome to undergo translation and form proteins and ensure that the prokaryote can function properly and live.